Professor Bharta Majumdar, Professor Vibha Chauhan, Professor Tirthankar Bhattacharya, and all the other organizers, plus dear students. What I'm going to talk today is something which is actually you're not witnessed in the bacterial world, but it is happening all the time. The evolutionary arms race. But more importantly, this evolutionary arms race, which happens in every organism, has been taken advantage of. We scientists have developed technologies. These gene editing technologies, what we are so much talking about, are possible simply by doing simple research with the bacteria and the bacterial viruses. See the point. So many major discoveries have been made, Nobel Prizes have come, and that has led to technologies. So for students out there, you can do extremely simple experiments and you find some phenomenal things, and then many of these things can be taken into new technologies, and which has revolutionized actually biotechnology. Modern biotechnology, what you are talking about, nothing but revolutions, couple of revolutions which I am going to speak today, which has led to finally now gene editing, what we are speaking, and it is today's title. Right. What you people already know in your class, I'm primarily trying to address the students, and my apologies for the distinguished members attending here. What you all know is you have seen it all along, around your gardens, outside, in parks. Every in animal kingdom, in plant kingdom, there is always a fight happening. There is a conflict happening all the time, and these conflicts are of so many and so many defense mechanisms, either through chemical means, but then of course you have a big swarming pair of fishes to counter the other larger components or others. Then they develop weapons, and we humans, Homo sapiens, end up using all kinds of ammunition to do the fight across and develop newer ammunition. So this arms race continues. But I am not going to talk about all these things. All these things you know about. Why should I bother wasting your time by talking about it? I'm going to speak about arms race, which has led to the development of phenomenal technologies in the next 40 minutes. What you already know is, perhaps you know, is there are more bacteria than any other part of life, whether it's plants, animals. Look at it. Sometimes when you ask students, they say insecta is the largest group. Insecta is one class in the family Arthropoda in invertebrate zoology. But then you have so many plants, algae, fungi, bryophytes, pteridophytes. They all come within the plant system, and then animal system. They are all here. The bacterial one is the vast one, and actually even there you have conflicts, and there are defense and survival strategies. And we are going to study very few of them now, relevant for today's presentation, relevant for how that has enabled us to develop new technologies. So bacteria have their enemies, not always enemies. They sometimes love to stay inside the bacteria forever. So these are bacterial viruses. They come in so many shapes and sizes. That's not important. Important thing is, there is an arms race between bacteria and bacteriophages. I'm not going to elaborate this slide. Basically to say, when bacteriophage, which is this head and tail, it infects the cell, then the host tries to come up with alternate mechanisms. So it prevents the infection. Then bacteriophage develops new mechanism, able to infect. Then there's another new mechanism from the host, prevents the infection. I'm not going to give these details. When some of you join for PhD, I'm going to explain microbiology class, all this, what this means, all these proteins or whatever it is means. So this arms race has been going on much before other species have evolved. So this has been going on all along. But but what I need to tell you right away is, despite defense mechanisms of bacteria, phages outnumber bacteria by an estimated 10 to 100 fold. Right now, it's estimated there are about 10 to the power of 19 bacteria in the universe or in Earth. I think mainly in Earth, I think, and there are about 10 to the power of 21 different bacteriophages. So there is a 10 time to 100 time bacteriophages. How it is estimated, etc., is something I will not elaborate. But in spite of this arms race, they coexist. There are obviously because they have defense mechanisms and counter defense. Simply, they find a way of survival. I hope I made this point clear. So there are many mechanisms then why bacteria adopt. They prevent the bacterial virus phages absorb, block the phage receptor, production of extracytoplasmic membrane so that they can, or production of competitive inhibitors, or say something called laboratory infection system, I will not elaborate, or prevent the phage DNA entry. And this is what we are going to talk today. There are two mechanisms to prevent the DNA which is entered to be destroyed. One is the innate immunity system of the bacteria, that is restriction modification system, and other one is adaptive immunity system. Adaptive immune system is in very much, this CRISPR is very much talk of the town these days. You hear about it all the time, gene editing and CRISPR, and we're going to spend most of the time there. But before that, because we are talking about arms race, because working with the RM system has led to phenomenal findings, and early gene editing was relying on RM systems. And since I like RM systems and they have laid the foundation for CRISPR work, I have to speak about RM systems. So suffice to say, RM systems are innate immune systems, whereas CRISPRs are adaptive immune systems. Why one is innate and one is adaptive, it will be clear to you. So in other words, we talk about immune systems in higher organisms, but trust me, in bacterial world, there are different kind of very primitive immune system, which is basically enzymes which target incoming DNA, and these are of different flavors, different types. And we will get a little bit of flavor, because obviously in the limited time I can't spend too much precious time of yours. So the phage infects then, DNA is unmodified, that is targeted by restriction enzyme, and the host enzyme is protected by the methyltransferase, which is so that the host enzyme is not replaced by restriction. Restriction enzyme is nothing but a biological scissor, molecular scissor, which cuts the incoming DNA, and modification enzyme protect that DNA. However, some phages escape that, and when they escape, this modification enzyme modifies it. So next time when the same phage infects, now restriction enzyme cannot work. This phenomenal discovery way back led to the Nobel Prize for 3 people. You are going to see some of them. Two of them are still alive, and you are going to see their picture in few minutes. Stay tuned. Right. RM system, restriction modification system. You all know about it. I think many of you do these practicals. You heard an enzyme called EQR1, which cuts sequence GAATTC. Find and at the palindromic site. The palindromic, they read the same way, top stand, bottom stand, Malayalam, or Madam, I am Adam. Either way, you will read the same. If you turn it around and they cut there, and as a result, the incoming DNA is cleaved. But the methyltransferase modifies this DNA so the restrictions then cannot cut. That's as simple as that. So there's a basically bacterial host defense just invading foreign DNA. But as I said, if the phage DNA escapes, it gets modified, right? So how I'm going to go through this slide just to tell you how co-evolution of defense and counter-defense mechanisms have taken place, taking an example of a very well-studied bacterial virus called T4. T4 infects E. coli. T4, ancient T4, don't ask me how ancient. The modern day T4 looks very different, but in laboratory you can prepare this ancient T4, which has got in the DNA cytosine, like any other DNA. When it infects host, incidentally, restriction enzymes come in so many types. Type 1, type 2, type 3, type 4, many types. I'm not going to spend much time. Some of you can look at up my review or other reviews. There's so much written about, textbooks will tell you. So the ones which are used in genetic engineering is mainly type 2 enzymes. Gene editing you use type 2 enzymes. And I'm not going to talk anything about it. Just about the arms race here now. The T4, ancient T4, when it infects E. coli, you have the type 1 enzyme, and that cuts the incoming DNA. So phages are not able to multiply. What phage did then is modified all its cytosines to hydroxymethyl cytosine. Anti-restriction system, counter-defense strategy. Now phage is happily multiplying. Then what host did, green rectangle is a host, bacteria, developed a new kind of restriction system which recognizes modified DNA, methyl-directed restriction, type 4 restriction system. So again, phage is not able to multiply. What then phage did is developed, added glucose residues to DNA. The modern phage has hydroxymethyl cytosine and T4. The modern T4 has hydroxymethyl cytosine as well as glucose added. It multiply. So this I'm not going to discuss this. Like this, to solve this law, again, if some of you join for microbiology, if I'm still teaching, I'm going to teach what these are. It's not really important for. So this arms race has been going on. And I'm going to tell you a little bit of what I showed for arms race. But then, people who discovered restriction enzymes, so it is Werner Arbor who gave explained the mechanism of restriction modification. He said, restriction enzyme is the one which degrades incoming DNA and host DNA is protected by methylation. He said restriction modification. He gave the terminology. Then Hamilton Smith and Daniel Nathans, unfortunately, Daniel Nathans passed away a long time ago. And this is not Daniel Nathans. This is only Partha knows who this is. And, and this is Hamilton Smith, the other Nobel Prize winner, who purified restriction enzymes. Nathans and Smith purified the first restriction enzymes, type 1 restriction. Then there is an explosion of research, and you have type 2 enzymes. And in fact, my own lab, when I started, the first biotechnology company, the very first biotechnology company, Bangalore Genie, we cloned a large number of restriction enzymes. We identified new restriction enzymes. This is a bacteria from rice fields called Oscillataria, originally called blue green algae. Actually it's a cyanobacteria, and from there we found restriction enzyme. Then we found a restriction enzyme, the Streptococcus thermodiasticus, and all those are not left. Point is, the first biotechnology company cloned enzymes, and they are very happy afterwards. They have been making money, and that's not all. So we thought we should also not only do clone and do, we should do some basic research on type 2 enzymes, more simple enzymes. What I'm showing here is, see, all of us studied restriction enzymes, and you people have studied in your class now, like this. Enzymes are supposed to be site-specific. They cut at the predetermined site, as we already discussed. For example, this particular enzyme has to cut at the sequence called GGTCC. So there is a plasmid, it has got only one site. But if you use more enzyme or magnase, we see multiple cleavage. So there is one site. So simply this enzyme has a promiscuous activity. And I will not, but then we made mutations. I will not say we engineered the enzyme, high fidelity. and that is a world patent. How did I not say this? Published here. Basically, the world patent. We made this. This is important for the study of what I'm showing for arms race. So wild type enzyme which cuts more and more incoming DNA, whereas wild type is specific, will not cut as much. Right. So basically, it shows that by making point mutations, you can change the specificity. And if you have other ways of doing it, long time ago, in a paper in publication in Nature, I showed that there's a sequence, the enzymes are evolved by recombination. Right. So promiscuity is not an exception. That the NEB, largest restriction enzyme manufacturer, had high fidelity enzyme based on our discovery. If you go to the website, they say work done in English science, and now they sell enzymes for high fidelity enzymes. So what I'm going to show you now is that this promiscuous behavior is advantageous for targeting the invading foreign genome. So this is basically a mechanism of arms race, and what you simply, this another paper, what we show is you use either wild type enzyme which cuts in additional sites or a high fidelity enzyme. Basically, now incoming phage gets more degraded here compared to here, because methyl transferase modifies only species. So, the covalently arms race, as I said in the beginning, anti-restriction strategies ensure that there are more phages. Phage DNAs are modified, and you already gave the example. And they avoid these palindromic sequences. There are enough examples. So now, if you have such a promiscuous activity, even the phage mutates the original restriction site, promiscuous activity gives extra advantage for bacteria against the phages. There's one situation where bacteria is getting an upper hand. And this is what is shown here. In fact, when there is no RM system, you acquire RM system, don't allow phages to bacteria, allow the phages to multiply, but then anti-restriction strategies allow the phages to multiply, then promiscuous restriction enzyme is an advantage. And what I also show you here is promiscuous, if you have it, then it also can bring in apoptosis of bacteria at late stage of, so in sometimes phage infection, then bacteria lysis, and basically as a result, self-lysis to prevent spreading to the next bacteria. Usually phages infect the host, and when they infect the host, they multiply there, and then they come out and infect the neighboring bacteria. But if the bacteria dies, then benefits the population, so it provides nutrients to surviving population, even if there is no bacteria. I can elaborate that if there is a question answer session. So basically, restriction enzymes, innate immune mechanisms, plenty, different classes. However, phages have found a way of overcoming restriction system. The innate immunity is good, but the innate immunity is not good enough. In fact, that's why enzymes in innate immunity start doing other functions. This is what we have proposed on many cases. And in the genetic engineering, in fact, gene editing, I will give the example as you go along. I will not elaborate on restriction enzymes have been used in gene editing. In fact, Srinivasan Chandrasekharan, and a professor in Johns Hopkins, was a postdoctoral student of Hamilton Smith, one who got Nobel Prize for the isolation of restriction enzyme. So he went on to prepare zinc finger nucleases, basically hybrid of zinc finger, zinc finger protein and restriction enzyme, which is used for gene editing and genome editing before the discovery of bacterial adaptive. So gene editing technology has been there last 15-20 years. It has been efficient, but the CRISPR adaptive immunity has made it more efficient. And that's what I'm going to speak in the next 15-20 minutes. Right. So what is bacterial adaptive immunity? Bacterial adaptive immunity is, you have the host restriction systems, that's the innate immune system. Then the phage infects now, If you don't have this CRISPR system, I will elaborate what is CRISPR system. Phages simply multiply and cell death. However, if you have this CRISPR system, phages are suppressed. You end up getting viable bacteria. So this is a global response against foreign DNA and in this case even RNA, when RNA phages, specifically sensitive to single nucleotide differences. Importantly, it maintains memory. That's how it is called adaptive immunity. If you remember, the adaptive immunity, the circulating antibodies are generated because of the adaptive immune system. It is produced from B cells. So basically, it is a memory. How this memory is brought about, I'm going to discuss. Why am I spending this much time on the fundamental discoveries? Because the genetic technology is resting on the shoulders, resting on this foundational research. These are simple experiments any one of you students out there can do, and these experiments have led to phenomenal discoveries. That is the reason, right? So, what are the CRISPR system? It's very simple. CRISPR, clustered regulatory interspersed short palindromic repeats, and these are those repeats. I'm going to elaborate. I'm going to spend a little bit more time here because this is the main component of gene editing in the present day context. So what we are doing here then, CRISPR system contains a large cassette of gene in the bacteria. It is through RNA prepared from this cassette. That's why it's called RNA-guided genetic styling system. And remember, it's a part of the adaptive immune mechanism. It's part of the evolutionary arms race. So CRISPR arrays are nothing but repeats, as I show, and spaces. Spaces, all in black here, but it is going to change in the next slides because there's like, and each one of them are different spaces. Then these are the genes which are needed for the whole working of this CRISPR-Cas cassette. So there's a leader sequence, and what it does, I'll come to that. And the Cas is CRISPR-associated genes. Many genes are there, and they have enzymes to cut the DNA and sometimes RNA as well, and associated proteins to do different functions. Right. So, how does the CRISPR-Cas work? Incoming phage, when it comes, viral DNA is exposed, or a foreign plasmid enters through conjugation process. You know what is a conjugation process. It's a simple microbiology way of transferring genetic material from one bacteria to another. And if there's time, if anybody wants more details, you can read up or you can ask me. I can spend hours talking about conjugation as well. But here, so the CRISPR complex basically, or the incoming DNA will be degraded by restriction enzyme, then this Cas can be targeted. Basically, what happens is short piece of DNA from this phage or plasmid gets incorporated into the cassette. That's why this cassette can go up to 50, 60, 70. There's the same kind of a spacer, but repeat, same kind of repeat, but the spacer every time is different. Every time a phage infects, this phage or phage X, Y, Z, A, B, C, every time some spacer can be incorporated. That's why 1, 2, 3, 5, and the next one gets incorporated. Now, this is called adaptation, or also called spacer acquisition. You'll keep hearing these words again, and you'll find it out anyway if you go through some reviews or some books. Immunity. So next step then, acquired immunity against subsequent viral infection. Don't forget this piece of DNA is from the phages. Each one is from different phage. So when that guy comes to infect again, what happens is CRISPR-Cas now make this into transcribes, make these small fragments from our CRISPR RNAs, RNAs go and bind to the incoming genome and inactivate. And basically, the incoming phage DNA gets inactivated by targeting, by interference. Something similar to higher organisms we talk about. RNA interference. Very similar. We sometimes talk about such interferences only in higher organisms. In bacteria, it's a routine affair, actually. Right. So this is a very quick slide. I'm going to say in CRISPR-Cas also, so much research has gone in, and that has led to type 1, type 2, type 3, variants of these, so many CRISPR mechanisms. I'm not going to discuss this. If you are going to do research on CRISPRs, you need to know this. There are a few groups working in India also doing research on it. And one in IIT Guwahati, one in IIT Delhi. So I'm not going to, one in IGIB. And so I'm just going to say there is variety. Remember, restrictions are your type 1, 1, 2. Simply, bacteria wants different strategies. That's what different ones. That's why you make different kinds of aeroplanes, make different kinds of arsenals. Basically, different weapons against the invading devices. So, coming back to the CRISPR pathway, because this I'm going to be repetitious simply because I want to understand this. I hope you will understand. If anyone of you don't understand, you can set up another Zoom call. I am here to talk to you so that you understand the basic concept, how gene editing is done. There are many scientists, top of the world, in leading institutions, they talk about CRISPR-Cas without knowing how they function. That's the wrong thing to do. Since they don't know, they assume things, and if you know, you can do the experiments much better. And that's important for all of us to study well. Your physics, your chemistry, your biology, if you study well, you will climb the limitless ladder of learning science. And when you do that, what happens, I'll tell you in the very end. You have the CRISPR pathway, you have adaptation, acquisition, and integration of foreign fragment to the CRISPR locus. Remember, these are same repeats plus new arrays. Repeats are in black here and different arrays coming from different foreign DNA, age or the plasmid. And this is from in the cassette. So then in the next biogenesis, you generate these small RNAs in the cell. You have all different RNAs made, and then in the defense, when the same phage comes, you have this piece memory circulating, and this binds, and then this enzyme targets it and cuts this DNA in here. So this is the adaptation, biogenesis, and defense. Extremely simple. But now we are going to use it for technology. How? I'm going to tell you. So now, unfortunately, this is the kind of talk I prefer to have one-to-one interaction. Right. So CRISPR-Cas9 mediated interference is a bacterial adaptive. We said that. We already talked about this cassette. The cassette has Cas9 enzyme. It's in type 2 CRISPRs. Then you have other genes. They're all needed for all this. There is variety of process, right? First of all, invasive phage, when it comes, when the phage or plasmid enters, one small piece, degraded piece gets integrated, and that becomes a new space, as I said. And they have the same set of repeats here. Repeats are in diamond shaped, and the rectangles are the spaces. Every time phage comes, new space. And the spaces can be large numbers, and the length can be varied, and there is so much written about it. Read up if you're not interested. That's not the business end here. We are interested in seeing how it's going to do gene editing. So this is important to know. So this is how acquisition. Then up in the leader region, you have a RNA made during transcription or CRISPR biogenesis, and this is called tracer RNA. Actually, it's a trans-CRISPR RNA. Trans-CRISPR RNA. I will come, and that is what is shown here. Trans-CRISPR RNA. What is it? I'll tell you in a minute. And then pre-CRISPR RNA, because when this is transcribed, as I showed before, you'll end up. This is a polycistronic RNA made transcription, and this trans-CRISPR RNA is complementary to this repeats. In fact, that's why it's transcribed in the opposite It's the same kind of sequence, so it's complementary. So trans-CRISPR RNA, tracer RNA is complementary to pre-CRISPR RNA at specific spatial sequences. Specific repeat sequences. Repeat sequence, different spaces are acquisitioned every time. Now you end up using maturation by RNase 3 and the other enzymes. They trim it, make it CRISPR RNA ready for silencing. So this one, Cas9, binds to this, and now this whole complex, Cas9 makes the cut, the introducing new phage comes DNA, and this, remember, this is the spacer which is obtained, and that's where this cut occurs. Okay? And these are the fundamental discoveries led to Nobel Prize. Same thing I'm showing it another way. This is a foreign DNA, CRISPR loci. You end up acquiring the new spacer. Then you have the tracer RNA, as I said, trans-CRISPR RNA like this, and that gets assembled, CRISPR RNA, and this kind of sense antisense complex is assembled because that's how you guide it into the enzyme and target the incoming DNA. So there is a complementarity here and the base complementarity. Watson. What made myself clear? I'll be happy to elaborate further. So, just flashback. Because I am handicapped because of time. I said before CRISPR was done, Hamilton Smith student, Srinivasan Chandrasekharan, in pioneering work, he developed the zinc finger nucleases. Then there is the INT-encoded meganucleases. They are used for genome editing. All successful efforts. And then you had talents. Talents are again restrictions I'm fused. This is a type 2S restrictions I'm fused with. This is Srinivasan Chandrasekharan work. He's done many other things. Right now he's working on CRISPR. John Hopkins guy, a professor, and great guy, a good friend of ours. And then talents also inspired by the zinc finger nucleases. All this do the genome editing. So is the CRISPR-Cas. They all do the same thing. So what they do? However, CRISPR-Cas9 has become the preferred tool. It always happens when the new model comes into market. You are like most of you, I mean, almost every one of you have fancy iPhones or smartphones. You end up getting a newer model which has much more facilities and operational easiness, and that's what is CRISPR-Cas9 is all about. It has so much easy way of doing gene editing compared to any one of them. But these are all very good. They laid the foundation. I can't forget it because simply I gave the idea how to develop these things. So when you make, then what they do, interested where you want to do genome editing, you introduce double-strand break in all these systems, and then you have a process called non-homology end joining, NHEJ. I will not elaborate this mechanism. Any textbook will elaborate for you. Or homology-directed repair, which is basically a recombination process, which will allow insertion. This will allow indels, deletions or insertions to be made, so you can disrupt the gene by this. Or you can introduce a new gene, or even find a way of deleting the gene. Right. So now we have no business. What we studied so far, or what we learned so far, I hope you learned, is the basic fundamental discovery. And many people contributed for that, but only 2 people got Nobel Prize. I'm going to make a comment on it in a minute. Stay tuned. But what you see here then is, what I said already. Integrate exogenous DNA, spacers, repeats are on the same space, and transcription, you end up getting tracer RNA, and then CRISPR RNA, and assembly. But this way, you can use it for doing genome editing. But you have to do a little bit more tricks to do genome editing. That's what we are going to discuss next. But before that, I must tell you, the enzyme which is doing this cutting is a fascinating enzyme. Structure has been solved, like many other restrictions. It's a recently evolved enzyme. Why I say it's a recently evolved? Because all of you, your madams, your teachers, taught you about enzymes in your biochemistry class. Enzymes do all kinds of work, and restriction enzymes also do the same thing. And they cut the enzyme, they cut the DNA, but they all have a single active site. Because active, but in the restriction enzymes, you have about 5-6 different active sites. So we have discovered totally new active site, highly cited paper, but that's not the point. So you have so many different. Each enzyme has its own active site. HNS is Cas9, CRISPR enzyme Cas9 has 2 active sites. One active site called HNS active site. In fact, this one we discovered for type restriction enzyme. Now many of them. And in my lab, Saravanan, who is a professor in IIT now, he discovered that. And then another active site is called RAW-C. It's found in recombination enzyme in E. coli and other bacteria. So 2 active sites are borrowed in this evolved enzyme. It's a fused enzyme in which 2 different active sites. One strand is cut by one active site enzyme of the enzyme, other strand is cut by other active site. 2 different active sites. In case of enzyme engineering in nature, this has happened because don't forget adaptive immunity. That's the reason CRISPRs have evolved much later, because bacteria had to find a newer mechanism to counter the fetus. And that's why these evolved. Now, as I said just a few minutes ago, this is the assembly, and this assembly is needed for genome targeting, but this is a bit more cumbersome. You can make use of it even now, people use this way. But even better is to use this to make a synthetic DNA in which you have the spacer and you join this together. Spacer RNA and CRISPR RNA fused to basically make a sgRNA, sgDNA. But that is what is shown here, a linker loop and tracer RNA and CRISPR RNA joined, and then so it all comes together. After all, it's able to assemble like this. Then it's much more efficient, but you can do it even without that. So basically, you program the whole thing for genome editing. This is what Jennifer Doudna's postdoc did. It's not only Jennifer Doudna's, Manuel Charpentier's student did, and importantly, another person, Varghese Sisnis, did that, and they all did Carpentier and Doudna did together, or they did independently, but then they collaborated. But basically, the process, we have this dual active site enzyme Cas9 is able to target the pre-designed one, because as I said, you can pre-design the sequence where this complex can align. You can design it to go exactly whichever region. It doesn't have to be any more incoming phage. You can do the genome editing by this technique. And basically, the result you can direct with non-homologous end joining to gene disruption by small insertion or deletions, indels, or you can introduce a donor DNA, gene correction or insertion. So if you want to do gene therapy, if there's inborn error, if there's a defective gene, you can go ahead. This is what done by other enzymes, and this is what is done by CRISPR-Cas. But CRISPR-Cas is much more easier the way I described that. So you can do in CRISPR-Cas, either you can do insertion deletion, genome engineering with Cas nuclease. So your donor DNA can be introduced by homologous recombination, homology directed repair, or you can do double clicks. You can have 2 different Cas9s target 2 different regions, and as a result, you can have double another way of recombining. So there are multiple strategies like this, or you can do gene activation using CRISPR. In this case, you use a mutant CRISPR enzyme, so it doesn't cut here, it binds, is fused to an activator, or if it is repressed for gene silencing. You can do gene activation, silencing, right? So you can do the labeling and you can visualize where the target of interest, your protein goes and binds, etc. To umpteen strategies, I will show you the examples of some of them. We can do multiple applications simply because you know how to edit it. You basically have scissors at your hand. It's as simple as just, and in fact, some of your students, you should be able to do it if your teachers teach this in your practicals, if you have the right one. But it's much more, I mean, you can even learn the simple restrictions and you can be in a cutting, you can realize the lab. Right. So this is for me, this one is also a hero. This guy, Verghese, you spell the second Verghese Cisnes, Virginius Cisnes. I simply call him Verghese. He's a good friend of mine. Pioneered the work on restriction enzymes a long time ago, solved so many structures, and he moved on to study CRISPRs. So what he did, so important, he developed his own gene editing technologies, but he's sitting in Lithuania, and he shared one of the very prestigious prize called Cowley Prize, along with Emmanuel Charpentier and Jennifer Doudna. You would have heard Emmanuel Charpentier and Jennifer Doudna got Nobel Prize in Chemistry this year. But this guy did as much, and if you go to the Cowley Prize website, I request Professor Partha Majumdar to look at it, especially Partha, simply because he deserved as much, if not more. In fact, his paper was delayed publication. I think I can. But Cowley Prize is very prestigious, almost as important as Nobel Prize. He shared with them, right? And these two got the Nobel Prize this year for the understanding the CRISPR-Cas mechanism and how it can be used for gene editing technology. Emmanuel Charpentier and Jennifer Doudna. I must say their work is very important, but I feel the other guy was ignored. So the next 5 minutes I'm going to summarize. I hope that's okay. Just quickly, I'm not going to spend much time because the CRISPR-Cas enzyme then can introduce into if you're doing a genome editing in mouse, and it has been done in so many cases now. So this enzyme is introduced, transits mouse DNA, and it targets the guide RNA. This is a recombinant. I told you, tracer RNA as well as and CRISPR RNA is fused this guide RNA, and the guide RNA identifies the corresponding DNA sequence. Two nucleases of the two active sites cut the DNA strands. Then you can do mouse DNA repair machinery makes mistakes, reconnecting, that is non-homologous end joining, or it makes correct replacement of the cut section with the new DNA. This is a homology directed repair or recombination. Embryos are grown in culture dish, then transferred into foster mother. A small sample of the mouse pups tissues tested for by polymerase chain reaction (PCR). You know what is PCR? So to see whether you have got the correct one. So every experiment one will need to check in the lab. That's important. So there are hundreds and thousands of papers. There is an explosion of science of gene editing. So just the titles I'm showing, targeting genome engineering human cells by Cas9 by the RNA editing. Enhanced efficiency of human footprint stem cell genome editing to replacing talents and CRISPRs. All publications, hundreds and thousands. Generation of gene modified mice via Cas9 gene targeting. Enhanced efficiency of human footprint stem cell and efficient genome editing in zebrafish. Zebrafish, bacteria, name a system it has been done, RNA-guided bacterial genome editing. And now this is a small representation of in the plant systems. Banana, cassava, cotton, rice, wheat. So everywhere this gene editing is done, gene disruption, viral gene, is to avoid the virus and promote a disruption. All kinds of things have been done. I don't want to go into details. It's an explosion of science by using this fundamental discovery. So this is one of my last slides. Gene editing is for gene therapy. What is gene editing for gene therapy? A way forward. What you do then is you reprogram somatic your embryonic cells. People who did reprogram embryonic stem cells got Nobel Prize while ago, Yamanaka Now, this I will not elaborate. This is a pioneering, phenomenal discovery. And then once you have embryonic stem cells, within them you do this CRISPR business. What I said already. You have the CRISPR-Cas9. You have made the right kind of gene editing. You have got the guide RNA which is assembled here and targeted region where the enzyme Cas9 cuts. Then you end up inserting. If you want a if you want a defective gene to be silenced, you do this. Or if a viral gene needs to be HIV gene to be silenced, you do this. Or then if you want to replace the defective gene and put the correct gene, you can do the correct gene insertion. Then take this edited. These are all happening inside these cells. Then take it into this cell, differentiate it, allow them to differentiate. Of course, make sure at this time PCR that you have the correct ones. Then you characterize the cell identity. Then patient match the healthy cells, insert. This is how gene therapy can be done. And CRISPR-Cas is only not that. It's gene editing, what's what we discussed. You can study for gene regulation, I described. You can have a defective Cas9 to study gene activation, gene silencing, or you can do the epigenetic study, or you can do visualization studies, or you can do single base additions, or you can do RNA targeting and chromatin, understanding chromatin structure function and chromatin imaging. And this is endless. There are much more. I can't, because if I speak more, Partha will be unhappy. Story thus far, then, what I said is there is a co-evolutionary arms race between bacteria and their viruses. I hope I have convinced you. This co-evolutionary arms race has been happening all the time. It has been happening, and there are newer mechanisms coming. CRISPR is not the end of the evolution of the bacteria. Most of us want to appreciate these bacteria. They are much more clever because all of us have more bacteria than our own cells. We have our body in the gut, everywhere. Those with hair, without hair, like me, all of us have too many bacteria on our body. You only end up clearing a small amount in the morning when you take bath. So the bacteria, all these bacteria, evolve. And when they are evolving, they also develop strategies for their survival. And restriction modification systems and CRISPR-Cas are their weapons against invading viruses. Viruses have evolved counter strategies. So CRISPR's restriction modification systems, there is evolution of anti-restriction systems. I can speak hours on this, but I'm not going to. CRISPR's, your anti-CRISPR mechanisms now. Your type 1, type 2 anti-CRISPR mechanisms. My own student, after finishing PhD, did fantastic work in the University of San Francisco on anti-CRISPR. Now, in the last 5 years, several new anti-phage mechanisms have been discovered. The amazing ones, published in Science, etc., in leading journals. These, who knows, they may be developed into newer technologies. And those new technologies, I may be retired, but still I will be looking at for these technologies. But in your lifetime, you will end up seeing much more path-breaking technologies than CRISPR. Who knows what it is? Science is then basically climbing the limitless ladder. What I have done is a small facet of the bacterial world, though it is a bigger world than what we know. There is an ever expansion of defense and counter-defense mechanisms. And in the process, bacterial phages learn to live, co-evolve, therefore developing newer strategies. These new antiphage mechanisms I discussed in my PhD class, but I'm not going to spend time now. They are complicated and also in the evening I should go you. But suffice to say, when you do this kind of work, you end up meeting eminent people. What you see on the left is Rich Roberts, a Nobel Prize winner on gene RNA splicing event. Splicing not by CRISPR-Cas or restriction modification. But now he heads the research in New England Biolab, largest restriction enzyme. He also worked on restriction enzymes, but he got Nobel Prize for splicing of the RNA, lipid RNA. And this, if you don't know who it is, this is Jim Watson of the Watson person standing in between, who it is, who took your 50 minutes of precious time today. And this is our own hero, and we claim our own hero, but he is not our own actually. He's the president of Royal Society. Not only president of Royal Society, he's the Nobel Prize winner for ribosome structure. He's smiling because he had idlis in my house. Venki Ramakrishna. What I'm trying to say is, I have enjoyed doing science, and I have enjoyed talking to students about science. Unfortunately, it has to be in this forum, and in this forum is not the best forum. And I have come to Delhi College's Venkateshwara. They invited me. I've done lectures. And next time, after the COVID is over, perhaps I can see one-to-one students there, and then I can answer questions whole day if they have questions with the next. So pray for me and for all of you to see that this damn COVID is gone away by the splendid work of others. But meanwhile, what I shown you is, even if you don't get Nobel Prize, and obviously everybody cannot get Nobel Prize, you end up getting to talk to these Nobel Prizes and learn from them, and it's nothing, inspirational way. So do some research, climb the limitless ladder. There's nothing like you learn everything and enjoy science. Thank you for your attention.